is Razvan. I'm a PhD student in the On-Round Lab at Simon Fraser University in Canada. And today I'll be presenting a selection of an RNA polymerase ribozyme with a clamping domain that facilitates processivity and promoter recognition. This work was just published in Science 2 weeks ago. The RNA World hypothesis proposes that early in the evolution of life, RNA could both store genetic information and perform catalysis, functions which were gradually replaced by DNA and enzymatic proteins, which are more stable and efficient. Central to this hypothesis is an RNA polymerase ribozyme, or an RNA replicase, that is capable of both self and general RNA replication. If a prebiotic soup could have emerged containing NTPs, different kind of polymerase ribozymes and their corresponding genomes or related genes, indicated here by different colors, how did the concept of conditional gene expression emerge in such a way that a specific type of replicase could have efficiently used available resources to replicate itself and related genes, but not wastefully replicating every RNA it encountered, and also avoiding replicative parasites? This question gives rise to the idea of promoters and selective replication, which we see being extensively used in today's modern biology by protein polymerases. And also, would primitive polymerases evolve much like today's RNA viruses to prefer replication on a circular template to allow rolling circle amplification, replicating full genes independent of promoter location? The story all began with the Bartel and Szostak in vitro selected class 1 ligase ribozyme. It was isolated from a large RNA sequence pool. This ribozyme is capable of catalyzing the 5' to 3' phosphodiester bond formation of two RNA molecules, shown here in red between the ribozyme itself and a short RNA oligo. And the ligation chemistry for this ribozyme is identical to the chemistry performed by a polymerase, which is a nucleophilic attack from a 3' hydroxyl on a 5' alpha phosphate. With the class 1 ligase as a starting point, a second functional domain was then selected in the Bartel lab, and it was termed the accessory domain, shown here in green, and transformed the ligase into a polymerase that was capable of extending an RNA primer that was hybridized to an RNA template in a template-dependent manner. This accessory domain achieves this functionality by creating an NTP binding pocket. And this showcased the multi-domain functionality that ribozymes can acquire, much like the multi-domain proteins in today's biology. The example shown here is the B6.61 ribozyme. It was evolved in our lab, and it's able to extend a radio-labeled primer on an ideal template by about 20 nucleotides. However, this ribozyme has low processivity, and it dissociates from the primer template in between each nucleotide addition. Looking at modern biology, and in particular the bacterial DNA-dependent RNA polymerases, they achieve template selectivity and at the same time high processivity, which are both missing from the B6.61, by a two-step mechanism where an open form polymerase uses specificity factor, the sigma factor, to recognize a double-stranded promoter region on a DNA template, localizing the polymerase to the template, and ultimately undergoing a conformational change to a closed form, releasing the sigma factor, and transitioning to a highly processive elongation. So now, can we use in vitro selection to select for a third functional domain for the B6.61 ribozyme, a clamping domain that would simultaneously allow promoter recognition while also increasing processivity in a way that can be directly compared to modern biological polymerases? Similar to the protein polymerases, our ribozymes would adopt an open form, partially hybridized to a specificity primer, shown here in this case an RNA primer, and use this primer to recognize a promoter region on an RNA template by complementarity. Once localized to the template, the primer gets stripped off the ribozyme and fully hybridizes the template. And it causes this conformational change of the ribozyme to a closed form and allowing processive extension. To construct the selection pool, we started with the B6.61 ribozyme and added a fixed 5' clamping arm, denoted here as the primer binding site, which can partially hybridize to a short specificity primer shown here in red and black. To select for the second functional clamp arm, we removed non-essential sequences from the B6.61 ribozyme, and then added a pool of random nucleotides, 76 nucleotides to the 3' end, and resulted with an RNA pool that had a diversity of 10 to the 13th. In combination, these two arms should allow the conformational change to a closed form when the primer is stripped off onto a template, and ideally the clamp should surround a primer template. We used two selection schemes implemented over 30 rounds to select such ribozyme. The first was a clamping selection scheme, which targeted the ability of molecules to open and close several times around the template. Here, a primed open pool molecule were incubated onto a circular template that was immobilized on streptavidin magnetic beads. Functional ribozymes would localize to the template, undergo a conformational change from open to closed form, and would remain trapped on the circle while we can wash away non-functional molecules. Functional molecules were then released from the immobile support, either with buffer as a negative control or fresh specificity primer, triggering a conformational change back to the open form. And then to increase selective pressure, the whole process can be done a second time to force the molecules to do an open close twice. Quantified this recovery, shown here on the left. We can see the buffer recovery in red, and then the primer recovery in black for both the first on and off event and the second. And then we saw an increase from round 19 to 21, the last two rounds that used this particular selection scheme. The second selection scheme was targeted to select for both clamping and processive RNA polymerization. Here, open pool molecules were incubated on a circular template, and they were provided with NTPs, but instead of CTP, they were given a biotinylated CTP. This allowed functional molecules to clamp onto the template and then incorporate the biotinylated CTP into the RNA products being synthesized, which allowed the capture on streptavidin magnetic beads in a complex. The entire complex was then washed on the beads, and then the functional molecules that could go back to an open form were recovered with specificity primer. The selection was stopped after 30 rounds after observing an increase in pool recovery. Mutagenesis was performed between round 27 and 28, and the extension time was changed from 2 hours to only half an hour for the last 2 rounds. After 30 rounds of selection, we see a dramatic decrease in pool diversity, with 5 ribozyme families dominating. This decrease in pool diversity was inversely correlated with the observed pool activity, as we can see in this gel. The most abundant and highest functioning sequence was termed the clamping polymerase ribozyme, or the CP ribozyme, and we can see it here in a primer-bound open form. The 3' clamping domain could be truncated down to just 45 nucleotides, and it's predicted to contain 2 stem loop structures separated by a junction sequence, the junction sequence having the highest effect on activity when mutated. Covariational analysis proposed a conserved hybridization region between the 5' primer binding site and the conserved junction region of the 3' clamping domain, with the closed form looking something like this, where a primer template would thread through it, as shown in the bottom cartoon. Now, the CP ribozyme shows high extension on 2 different circular templates, template 1 and 2. They were generated from random sequences, and they have 2 different promoters, and they work with either primer 1 or primer 2. And we can see that we have about 85 nucleotide extension on template 1 and about 40 nucleotide extension on template 2. And most of the primer is being used up during these extensions. Now, this extension is directly comparable with other more recent polymerases from the Hollinger and Joyce labs. However, these labs use direct hybridization to get the polymerase localized to the template and to prevent its dissociation. And they also use repetitive sequence templates, so they don't use a clamping domain. The clamping domain is critical for effective primer extension, and we can see this by comparing it with the B6.61 progenitor on both template 1 and template 2. And we see that the original B6.61 can only extend about 5 nucleotides on the template 1 and no nucleotides on the harder template 2. And also removing the 3' domain off the clamping polymerase also kills the clamping polymerase's activity. Now, does this clamping domain offer true processivity? So when given a template, a highly processive ribozyme should be able to stay localized to its template and primer. However, a low processivity ribozyme, when given a template, can fall off the template, or it can fall off its original template and find a second template that lacks a primer, and either of these scenarios result in polymerization. Now, one way to measure processivity is to look at the extension efficiency facilitated by this clamping domain, and to quantify this efficiency, we defined and measured an extension ratio, or ER, for a range of promoter and templates. The numerator was defined as the extension by an open clamp polymerase that was given a template, divided by an extension of the closed clamp polymerase given a template that already had a primer on it. The extension ratio was measured by maintaining the primer and the ribozyme concentration in a 1-to-1 ratio, and they were titrated down over 2 orders of magnitude on a fixed high template concentration. As expected, the B6.61 progenitor had a concentration independent extension ratio of 1 on the circular template 1 due to not having a clamping domain, and we see here on a log scale graph. However, the CP ribozyme showed a 13-fold higher extension ratio at low primer polymerase concentrations with both a circular template 1 and the linear template 1, showing a stable association with the templates. Truncating the linear template 1 to a much shorter construct, short template 1, ST1, resulted in a low extension ratio that was completely independent of concentration. And this is consistent with the lack of a track for the clamping polymerase to hold on to. This lack of polymerization efficiency could be partially rescued by adding oligo A template sequence to the 5' terminus of the short template, or fully rescued by adding oligo A template to the 3' terminus, and also circularizing the short template with oligo A, showing that the clamping is template sequence independent since it's poly A template, and as long as a long enough template track is provided so the polymerase has something to hold on to. To further explore processivity, we immobilized the polymerase complex onto strip-tavidin magnetic beads, and then incubated with a range of templates and measured the off rate of either the primer or the template off the immobilized support. So when given a short template, the polymerase should not be able to hold on to it, so it loses both its primer and its template as it goes to a closed form on the immobilized support. However, when given a long template, it should be able to hold on both to the primer and the template. This is exactly what we found. The reverse complement of the primer 1 and the short template 1 nearly fully stripped the primer off the immobilized complex, while the primer was retained when using templates such as the poly A longer templates and the circular templates 1 and 2. Likewise, after 4 hours of washing, around 50% of the poly A templates were retained on the immobilized complexes, while only 1% of the shorter template. Lastly, a processive immobilized complex that has a sliding clamp should also be capable of driving polymerization around the circular template. And also it should be able to reach and extend primers found a substantially distance away from the promoter binding site by first extending the primer 1 and then sliding over to the 3' end of the second primer and extending it as well. This is what we saw. The bead immobilized open complexes were able to drive primer 1 extension around the circle. They also extended secondary primer 1+5 and 1+40. And we can see that when a second primer is in the way, the polymerase stops primer 1 extension and then moves on to the second primer and extends it. And these complexes were stable on the beads for at least 5 hours with washing. Now, having processivity, we return to the story of promoter specificity and gene selection. The clamping domain also confers promoter selectivity, depending on which specificity primer it is bound to. So CP ribozyme open complexes were formed either with the red primer 1 or the green primer 2. And they were able to be template specific. So with the primer 1 complexes, they were able to extend the red template 1, but not the green template 2. And this was true even when both templates were mixed simultaneously. And the same is true with the P2 complexes when it comes to the green template.
2 minutes, please.
Yes. Now, can we take this property one step further, where two distinct polymerases, CP1 and 2, can use a universally short primer, perhaps made abiotically, to synthesize a specificity primer with sequence unique to the polymerase. So we modify the 5' end of the polymerase to make two new ribozymes, each having a unique 2-nucleotide 5' tag, and both of them were able to extend a 2 or a 7-nucleotide universal 2-nucleotide or 7-nucleotide shorter universal primer, and they extended it to its full length using the ribozyme as the template. Now, using the self-templated primer extension feature, a tagged specificity primer can control promoter-based extension, allowing extension on the green type template while preventing extension on the wrong tag, the red template. And we see this in a gel where a full-length control green primer extends well on the green template, but not the red. And also, when a polymerase tags a universal short primer, it can extend well on the green, but not the red. So although these clamping polymerase ribozymes are not achieving self-replication quite yet, they do provide the architecture to overcome the remaining key hurdle, which is strand displacement via rolling circle replication, which is required to get the RNA products off the template that encodes them. And we have demonstrated how specific genes could be selectively copied by such processive clamping polymerases, with promoters determined directly by the polymerase's own sequence, and allowing, even in the absence of compartmentalization in an RNA world, a specific set of genes to be selected, as shown here with the green, and provide a mechanism to avoid replicative parasites. With that, I'd like to thank the audience, the funding sources, my supervisors, and our lab members. Thank you for your attention.